Good morning, students. Last class we discussed evidence of organic evolution. Mainly, evidence are coming from morphology, anatomy, homologous and analogous organs, vestigial organ, connecting link, and atavism. Today we will discuss what are the proof or evidence we will get from embryology. So we can see different animal, their embryo. You take example bird, dog, human, fish, salamander and turtles. So their embryos photo, just to compare it. Every embryo we can see similar shape and size. After the development of zygote, embryos, their shape, their size, their structure, all we can see similarity. So this is a one proof we will get from embryo. Not only embryo shape, we can see another very good example is a notochord. Every chordata or vertebrate, their origin stage, their embryo stage, we can see notochord or a backbone like structure. So this is a similarity. For same method we can see some temporary embryonic structure. In embryo time, gills are forming. Every embryo we can see gills, including human being. But after embryo change into newborn baby, this gill slowly disappearing. It is a temporary embryonic structure. From this 2-3 point, we can identify there is a evidences we will get from embryo. And another important theory is called the recapitulation theory. This is one of the greatest scientists, Ernst Haeckel. In 1866, he proposed this theory. According to this theory, ontogeny recapitulate phylogeny. What is the meaning of this? It simply means embryo in their development repeat the evolutionary history of their ancestor. So every time when we discuss about their evolution, we can see some similarities. Then again we can see some primitive plant like gymnosperm, cycas and gingo. Cycas plant and gingo plant, their sperms are flagellated. So it is a similarities. Flagellated sperms, and same time bryophyte and algae, we can see prothallus, their body structure we can see some similarities. So lot of evidence we will get from plant also. Another evidence we will get from cell biology and biochemistry. Take example ATP molecule, phosphogene molecules, hemoglobin molecules. So every living organism, including higher organism like mammalians, aves, reptile, etc., frog, amphibian, etc., we can see hemoglobins are present. And insect also we can see hemoglobin like structure, that is white in color, but we can see similarities. Same method cytochrome, molecular biology we can see. Blood group, human being blood group AB and O is present, but some apes we can see blood group A, B and AB. It is another similarity. RH factor is another similarity. Every living organism including human being, we can see RH factor. This RH factor we can see some rhesus monkey also. So from the cell biology and biochemistry, we will get lot of evidences. Another evidence we will get from molecular or genetical studies. So here protein structure, DNA structure, RNA structure, and another important point is a genetic code. Every human beings, every animal, every plant, there is a genetic code. Triplet genetic code is present. This genetical code also we can see some similarities. Human and apes, more than 97 to 98% their protein, DNA similarity we can see. So from the genetics also we will get lot of evidence. Then another evidence we will get from geographical distributions. So according to biogeography, the geographic distribution of life form on earth, we can see Asian continent, African continent, America, North America, South America continent, they all are single unit in ancient time. They are called Pangea, and thousands of years after some force of gravity or some force, this Pangea break into different continent. Now also we can see India's northern side, African southern side, we can see lot of similarities. For African continent, western side, and the South American eastern side, we can see lot of similarities. So same method, Indian elephant and African elephant, we can see lot of similarities. Now they are two species because of geographical separation. Same method, Australia, we can see some animals, example kangaroos, they are seen only in Australia because of geographical distribution. And same method we can see one famous Darwin finches theory. Darwin studied some small finches or birds, and these finches mainly seen in Galapagos Island. Galapagos is a small 20 small island. They are seen the northern side of 100-kilometer west coast of South America. There he seen different types of finches. These finches character, their shape, their size, they are matching with the South American birds, like different birds. So Darwin also proposed some evidence, and this evidence is coming from geology. So from geology, from morphology, from anatomy, from genetics, from cell biology, so everywhere we can see different types of similarity and difference. So Darwin finches, mainly we can say it is a adaptive radiation. What is adaptive radiation? It is a process of evolution of different species in a given geographical area. Galapagos Island is a particular area. So there we can see different types of birds, their character. So they are adjusting that particular area. So we can say it is a geographical adaptations. So these are the 5-6 points we will get from evolution. Thank you.